All right, we're going to take a look at Chapter 10, Part 2. And with Part 2, we're looking at some of the applications specific to recombinant DNA technology. So this is how genetically modified organisms are created. So we're going to look at some examples and some of the techniques that are used. Before we get into that, I want to point out a basic premise. It's important to understand how this is even possible. So when it comes to Mother Nature, there is a universal genetic code, and that code, being universal, allows for genes to be moved from one organism to another and encoded and decoded the exact same way, and that refers to transcription and translation. So that the genetic codes of DNA are universal, and that transcription and translation operate under the same principles. If you look back at Chapter 9, I gave you a chart that had the codons and the amino acids to go with those codons. And those are the same codes that occur in virtually all organisms. There are a few exceptions to my understanding. There's almost always an exception to every rule. But with keeping those out of the equation for today, pretty much every organism uses the exact same genetic coding. So it's kind of like every organism speaks English. And since the language is universal, you can take any sort of message from one organism and put it into another organism, and the message can be translated the same way. If this wasn't possible, these techniques wouldn't necessarily work. It's not to say they couldn't find a way to make it work, but it certainly wouldn't work the way it works as we know it today. So that is very important to understand. DNA is universal. Another way of looking at it, as it says down here, DNA and genes do not have a self versus non-self form of identity. Cells do. Our tissue and our cells, they have markers that determine self from non-self. We talked about that in the immunology section. But the genes themselves, they don't have markers that say, okay, this DNA comes from you, and this piece of DNA came from somewhere else. Once a piece of DNA comes into the cell just treats it like any other piece of DNA, whether it was there to begin with or whether it was transplanted artificially. Cells just really don't have a way of knowing, and so that's an important premise to start with to help you understand how this is possible. So ultimately that means that when we take DNA from one organism, we put it into another organism, the recipient organism, i.e. the recombinant, will take that and transcribe and translate it into a gene product as if the gene that was transplanted came from that organism itself. Therefore, any protein that's associated with that gene will be produced by the recombinant organism. Now, since proteins ultimately provide traits and characteristics, any trait associated with that protein will also go along with the transplanted recombinant gene. Okay, so you have the addition of a gene that adds a new protein to the cell, and the protein creates a new trait. Now, when I say the process is nearly universal, there are subtle differences, and those subtle differences might be like different dialects within the same language. So you might have slightly different ways of using the language. You might have more or less steps in the process, and these things can complicate the overall genetic modification. But ultimately, all life is speaking the same language, as I mentioned. So ultimately, while there are complications, the language is universal, and so ultimately that allows the process to work, although in a lot of cases it is more complicated than I might make it sound. An example is that when you're taking and trying to move eukaryotic and prokaryotic genes from one organism to another, the gene splicing that occurs in eukaryotes can complicate that. So if you recall from Chapter 9, eukaryotes will process their messenger RNA after transcription. So there's a step in between transcription and translation that occurs in eukaryotes that you don't see in prokaryotes. And so that is an example where the language, again, is still the same. It's not a different language, it's just a different expression of that language, so to speak. It's an extra set of steps. So that's an example where it's not identical, and it makes the process harder, and so they have to figure out a way to kind of manage those differences. Okay, so but ultimately the language is universal. So when you're looking at how recombinant DNA technology is often applied, it is in reference to the creation of a genetically modified organism, a GMO. Now, it's often a hot button word in today's world. People hear GMO and they think usually negative thoughts. Maybe that's not always true, but I think that tends to be a general consensus. A lot of people have a very negative perspective on GMOs. A lot of people want to outright ban GMOs. A lot of people avoid GMOs at the grocery store. And while there are some legitimate reasons for that, I think a lot of it is also a little hyperbolic, a little over-exaggerated. So a GMO they come in a lot of different forms. But really one way you can look at a GMO, and this is especially how the technology started, they were really being looked at as a miniature protein factory. And the first GMOs were bacteria and microbes. And what they were basically trying to do is get the microbe to make a protein that it wouldn't normally make, that would be beneficial to humans, or maybe other animals that are beneficial to humans, whether it's direct or indirect, but in some way that's beneficial to humankind. And so really, the idea is that you have all these proteins that are highly beneficial. As one example we'll talk about here shortly, the insulin protein, which is what we use to control our blood sugar. That's a protein. It's also a medicine. Insulin is used to treat diabetes, so it's an injectable form of a medicine, but it's a protein. So one way to get insulin is to convert bacteria cells into insulin-producing, quote-unquote, factories. Since bacteria don't normally make insulin, you have to add the insulin gene. And so at that point, you create a GMO. But really another way of looking at it is you've created a protein-producing factory, or in that case, an insulin-producing, quote-unquote, factory. So that's one way to think about it. The same application can be applied to the production of RNA products. RNA is not something we get into as much here, but just like protein can be a drug, or it can be some sort of beneficial product, the same is true of RNA. Genetic modification of living organisms started in the 70s and has been progressing ever since. At the beginning, most of the GMOs were microbial, bacteria, mostly a few fungi. And since then, starting in the 80s onward, they started modifying plants and then ultimately animals. And today we see all sorts of different types of all three types of these organisms being modified to create GMOs. Microbes were the first and are still the most common. Microbial cells are very small. You can grow billions of them in a small space. They are non-ethical. There's no ethical concerns. There's really very little regulation involving animal welfare, which you get into with animals. Plants have less animal welfare ethical issues, but they take up more space, more energy, and they're more expensive because of that. So microbes have been one of the predominant forms of genetic modification because of their ease of work, and because they were kind of the gateway into the more complex aspects that occur with plants and animals. So cheaper and easier, generally, is why microbes were used first. So the way it works with microbes, and this is specific to bacteria, there'd be subtle differences when it comes to things like fungi, for example. But when it comes to bacteria, the way you create a genetically modified bacteria is by actually manipulating the way Mother Nature works naturally. So, for example, when it comes to bacteria, naturally speaking, they will transfer plasmids back and forth between compatible forms of bacterial species. So recall we talked about in chapter 4, also mentioned in chapter 9, we talked about bacterial conjugation and the way bacteria can link up with the use of what's called a sex pilus or conjugation pilus and transfer plasmids back and forth. here you can see the plasmid being transferred from one bacterial cell to another. So hopefully you remember that, a little bit of a review from previous chapters. So in other words, bacteria naturally transfer these things back and forth. And a plasmid is just an extra piece of DNA that can carry a small handful of genes. So it's a transferable piece of DNA. So this already exists in nature. And what scientists figured out is that they could modify the plasmid to carry additional genetic information. You could even take out some of the other information that was already on there and add in different information. So you had this pre-existing transferable element, and scientists basically just modified that transferable element. So it would look something like this. You would have to do, first of all, there's lots of research that goes into this, years of figuring out what genes are, where they're located, how to get them out of the donor organism. So for example, if you want to take a gene from a human being, for example, and insert that into another organism like a bacteria, you have to first know all there is to know about the human gene itself. So there's lots of research that goes on behind each one of these processes, and so we don't really get into that. So I just want to point that out. Lots of research before any of this even takes its first step. So that isolated gene comes from all that research. Once you have it, you will start by cutting the bacterial plasmid using restriction endonucleases, and they will open up the plasmid by clipping two spots of the plasmid. From there, then, the gene of interest can be inserted, and using ligase enzymes, the DNA can be attached to the pre-existing plasmid. So then we closed up, and you now have a modified plasmid. So it's got part of the original bacterial information, and now it's got this new gene of interest from whatever scientists chose to put in there. And now the plasmid is allowed to just do what it does, which is to be transferred from one bacteria to another. The difference is that now, obviously, the plasmid is modified. So when a bacterium goes to transfer that from one cell to another, it's now just transferring an extra piece of information along with it. And ultimately, the bacteria does what it does naturally. So in this case, humans are really just kind of adding an extra little piece to the overall equation and letting Mother Nature take care of the rest. Okay, so it's a partly artificial and partly conducted from a natural standpoint. Now, there's lots of examples where this has taken place, but one of the first examples, and to me one of the more interesting, is how they use this to create human insulin. So I mentioned insulin already a little bit. Insulin is a protein. It is produced by a particular gene in the body. It's called the INS gene, and it makes a protein. It's transcribed and translated in primarily pancreatic cells to create that protein. So diseases such as diabetes, type 1 and type 2, are a consequence of basically not having enough insulin. Depending on the type, you're either not producing it or you're not producing enough insulin. So type 1 is actually a type of autoimmune disease where the body attacks its own pancreatic cells. And this usually happens at a young age. So because of that autoimmune condition, the body literally stops making insulin. And so you literally then have to supply that from an external source. With type 2, you become insulin insensitive, and you don't have enough sensitivity to the insulin that you do produce. So you have to add more insulin to get the same effect. Either way, you don't have enough insulin, and high blood sugars result from a lack of insulin. So with these types of diseases, then the treatment is, as most of you probably already know, what I'm talking about here, but just to clarify, the way you treat that is that you take insulin from an external source and you inject it into the patient. Now, early on, so just a bit of a history here, before the 1900s, it was really there was unknown cause of diabetes. They kind of had an idea that there was something happening. People were obviously getting sick and dying, but they didn't necessarily know why, which is the case in a lot of diseases at that time. It was around it was around the 1920s, I believe, somewhere in that neighborhood, maybe 1930s, that they figured out that insulin, or they figured out that diabetes was caused by a lack of insulin. So they were able to isolate the cause of the disease. And once they knew that, they figured out then how to treat it. Once they figured out it's a lack of insulin, they said, well, add insulin. And sure enough, it would work to treat it in most cases. So once they figured out the cause, they figured out the treatment, then they just needed a source of insulin. And early on, that source, this is all again prior to genetic engineering, prior to that, the source came from animals. They would actually go to slaughterhouses. They would use the pancreas of primarily from pigs and cows. I think other animals, like maybe horses, were used on occasion. And they would grind up the pancreas. And it turns out that animal insulin is similar enough to human insulin that you can extract it and inject it and get the same effect that you would have with human insulin, or a near effect. And it worked really well. The problem that was alleged is that occasionally you would have hypersensitivity reactions. When you're injecting an animal protein like that, occasionally the body can see it as a foreign substance and react to it. I think for the most part it was a lack of purification, that they couldn't extract the insulin from the other animal proteins that came with it. So when they were grinding up the pancreas, it was adding antigens into the insulin along with all the other stuff from the animal organs. And when you inject that, certain people would have hypersensitivity reactions, and this can get pretty serious. So there was definitely an issue with some people that couldn't tolerate the injections. The other issue that they alleged was that the supply was limited. While you had a lot of animals from slaughterhouses, there apparently wasn't enough to treat the growing number of individuals with diabetes. And part of it was based on projections also. They were looking at the number of diabetes cases rising consistently, and they were projecting a shortfall moving forward. And that's probably exactly what would have happened had they not come up with a new form. So diabetes, as you probably know, is a fairly common disease these days. So this was one of the first applications for recombinant DNA technology. They had identified the insulin gene, and they had been able to extract that and isolate it. So what they did then was they took that gene and they added it to a bacterial plasmid. And then they allowed the plasmid to be uptaken by bacteria in the lab. And it turns out the bacteria would transcribe and translate that gene into human insulin. It's kind of an amazing thing. The genetic code is all that matters. It doesn't really matter what the gene originates from. So when you give a bacteria a piece of information on how to make a protein, it just makes the protein. It doesn't question whether it came from its own cells or whether it's human or what species this gene normally comes from. It just like reading a recipe. It just takes the recipe and starts making it. And so the bacteria then were modified to start making human insulin. And just like we do in our lab, once you create the first culture, you can then make more cultures by simply transferring the modified bacteria to a new growth medium and allowing the cells to repopulate. And so just like transferring a loop full of bacteria to a new test tube and allowing that new test tube to grow into a new set of bacteria, new population, you do that with these modified microbes. And so once you got the first modified cell, you then were just in the business of propagating that and making copies. And the bacteria would duplicate themselves, and when they duplicate, they make more of the plasmid, and each cell then goes on to make more insulin. So within a short period of time, you ramp up that production to an industrial level where you have large vats of bacteria being used to grow insulin, produce insulin. And so from there, it comes back to the purification, where you have to then extract the insulin from the bacteria and then purify that. And apparently, you don't have the hypersensitivity reactions. I'm not sure exactly why that is, but part of me thinks, I'm not sure, I don't really know, to be honest, but I think part of it was at the time prior to the 1970s when they were using animal insulin. I don't think the purification was as good. I think today, I know for a fact today they still use animal insulin, and I'm not aware that people have large hypersensitivity reactions today. That there may be, but I think part of that whole issue was the purification process. Because once you make the insulin, once you have the insulin, whether you take it from an animal pancreas or whether you create it using recombinant DNA technology from bacteria, you still have to purify the insulin. And if you don't purify that correctly, you're going to have other stuff along with it, contaminants, so to speak. And so whether it's a bacterial contaminant or an animal contaminant, either way you could cause hypersensitivity reaction. So I believe that part of the problem was largely based on purification. So with the improvements in that, along with the production improvements today, from my understanding, you don't have too many issues from either version. So today you'll find both types of insulin, and if you look at the insulin, it says animal insulin. That means it comes from an animal, obviously. A lot of times it comes from pigs. If it says human insulin, or sometimes called synthetic insulin, then that means it came from this process right here, where it's created in a microorganism. Now, from my understanding, they've gone on to use things like yeast in place of bacteria, but they're still using a very similar process using microbes. So this is a quick overview, a little more detail here. You have the gene that's isolated from the donor, in this case a human cell, and then you have a plasmid that's also isolated from bacteria. So you have really the what's called the vector, what you're going to use to move the DNA, and then you've got the DNA itself. Okay. The next thing you do is you have to cut both of these using similar restriction endonucleases. And so what that does is it cuts the gene in the plasmid using a similar, really I should say an identical enzyme, and it makes it so that the ends where they're cut have compatibility. So if you look here, for example, you cut the gene using an enzyme, and it cuts at this particular sequence here, or GAATTC. And then you use the same enzyme to cut the plasmid, so it cuts in the same place, GAATTC, and then it's the reverse on the other side. And so later then, when you take the donor gene and you go to insert that into the plasmid, they both have the same ends that are compatible with each other. And so they have complementary matching, and that gene will fit right into that plasmid and be now part of the plasmid makeup. So now you have a modified called a recombinant plasmid. And now you just take that plasmid and you allow it to be uptaken, naturally speaking, by bacteria. And really what that means is you just put it in a test tube along with the bacteria, and they will naturally uptake that plasmid, and then they will start to transcribe and translate. So this is what's happening inside the cell here. The donor gene in the plasmid goes through transcription. You create mRNA that goes through translation. It creates the protein product. That then is again upregulated. You culture more and more of these insulin-producing bacteria, produce larger amounts, and then eventually you get into the purification process where you extract the insulin from the bacteria. And that, I don't get into how the purification works, but that's another process in itself there. Okay, and so this is going through those steps here one by one. I'll let you go through these at your own rate here. So I'm just going to click through here every so many seconds, let you write down the information, and if you want to pause this and go back over it, feel free to do so. Okay, insulin is one example. There are many other examples, and this table here, 10.2 from your text, gives some examples of some of the more common transgenic proteins that have been produced. So I'm not going to hold you accountable for these. You don't have to go memorize all these. They're just some interesting examples that we don't go through each one individually here. So that just is just to look at. It's not something you have to study. So that, in a real quick overview, is how recombinant GMO bacteria are created. So next we're going to talk about animals just briefly. Animals is something you could really do a whole lecture on, and it's very interesting, but just going to do a quick overview here. So with animals, it starts the same way. You have a gene of interest that's isolated. Lots of research goes into that. And one of the more common ways that the transgenic organisms are created, transgenic animals are created, mice being the example here, is that you actually inject DNA into a zygote of a mouse. And there's some interesting techniques that they go into this, but they'll allow a female mouse to be fertilized by a male. And then, unfortunately, the female mouse is killed, her eggs are harvested, and under a microscope, they actually do what's called a microinjection. Here's an image of what that looks like. Maybe you've seen pictures of this before, but they have a device that holds the egg. I'm not sure what you call that, but there's a device that holds the egg, and then you have a microinjector that literally injects a tiny amount of DNA inside the nucleus of the fertilized mouse zygote. And then that adds genetic material into the cell. Now, this process is somewhat random. Once the DNA is added, some of it will be uptaken by the rest of the DNA. So cells have a way of kind of naturally incorporating DNA into their cells. It's not a consistent thing, so it's just somewhat random. So some cells will take it, some cells won't. They usually inject a large number of copies of the same gene. So they don't just put one piece of DNA, they may put several thousand pieces. And in some cells, you may only get a small number of those copies uptaken. In some cells, you might get large amounts of those copies uptaken. So the efficiency is somewhat random. Some cells will take up very little, if none. Some will take up lots. And so there's a lot of trial and error. A lot of cells that go through the process won't be successful, and there's a lot of discarded embryos and zygotes as a result of that. But there's a certain percentage of success done correctly over a long enough period of time, and eventually you get an embryo or a zygote that becomes an embryo that has just enough of the DNA to make it work. That modified zygote starts to become an embryo and is then transferred to a surrogate mother. So now the modified zygote is transplanted into another mouse, and the mouse now gives birth to the babies that result from that, just like it would any other scenario. Typically, you now have to study the mice and look for which of those actually has the modified gene. And not all of the mice, theoretically, that are born will have that. So there's a certain percentage that will have the modified gene and a certain percentage that will not. And that all takes later steps to determine all that. There's also other mechanisms by which this is done. They use viruses as another way to inject DNA. Retroviruses can be used to transport the DNA into a cell. And there's a few other techniques that I'm less familiar with that are less common, but there are, I think, 4 or 5 different ways that DNA can be put in. This is the most common, though. The next most common is the use of viruses, and the virus acts as the mechanism to bring the DNA in, and they use viruses that naturally integrate their DNA into the cell. So you modify the virus first, and then you allow it to add new DNA to the cell. So that's in a nutshell how recombination works in animals, and they've gone on to create a variety of different animals. Most of them are done for research. A lot of mice are modified for research in reference to different diseases. So they can study different types of cancer, they can study different types of bloodborne diseases, different types of genetic diseases. And what they're able to do is look to see how the addition of a gene affects the mouse, and then how different drug treatments can maybe affect the outcome of that disease. An example of where this technology is used in mice is a type of mouse called a knockout mouse. And these are mice that are modified in a variety of ways to create research subjects. And they can replace or knock out an existing gene, and that allows them to alter the genetics in somewhat of a predictable way that gives them the ability to then create research model subjects like the mice themselves that can be used for various diseases. This one here mentions research involving things like cancer, obesity, heart disease, diabetes, arthritis, etc., even Parkinson's disease. I've seen articles referencing Alzheimer's, cystic fibrosis, I believe it was sickle cell anemia is another one. So lots and lots of things can come from this, and they ultimately use a version of this transgenic modification to create these mice. And once they have them, they can use them for various research models. So a very important part of today's modern research involves transgenic mice, specifically the knockout mice. Okay, next we're going to talk about transgenic plants. And so this comes from a technology in which they can modify plants using a particular type of plasmid that comes from bacteria found in the soil. Now there are other ways of modifying plants, so I'm not going to cover all the different techniques. There were older methods that actually involved like literally shooting the DNA into a plant cell, and that's still used from my understanding, but less common. This technique here is, from my understanding, a little more commonly used. So the way it works is based on the discovery of a type of bacteria that naturally infects plants. Yeah. So this is, once again, a modification of Mother Nature. So there's a group of bacteria, a genus of bacteria known as Agrobacterium. One of the more common species used is called Agrobacterium tumefaciens. What these bacteria do naturally is they infect plants with a special type of bacterial plasmid called a TI plasmid. And that stands for tumor-inducing plasmid. So it's an odd twist of Mother Nature. And somewhere in evolution, these bacteria, which again naturally exist in the soil, they evolved plasmids that could actually be transmitted into a plant cell. And the plasmid would work in a very similar way that it works with bacteria, being transmittable genetic information back and forth. But in this scenario, the transmission of the DNA goes from bacteria to plant. And it apparently works when a plant, typically the roots, are damaged. And this could occur through parasites like insects, or it could occur through just natural damage. But apparently the cells or the tissue have to be somewhat damaged in order for the DNA to get in. So that's usually one of the first steps, one of the first things that has to happen. So a damaged plant cell is susceptible to this type of infection. And what the bacteria does is a very amazing thing. It inserts this plasmid into the plant cell, and the plasmid is naturally uptaken into the DNA of the plant cell itself. So the genome of the plant cell uptakes the plasmid DNA. And from there, then, the DNA of the plant is naturally modified. And apparently why the bacteria do this is that they modify the plant to start producing nutrients that it will then excrete into the soil that benefit the bacteria. That's my understanding of why it's thought these bacteria infect the plant in the first place. So they're basically a bacteria that modifies the plant around them to benefit them with growth factors that the plant wouldn't normally produce. A consequence of this is that occasionally the plant will grow a tumor that results from the infection. Here's an example, sometimes referred to as crown gall disease. These will sometimes occur near the base, they can occur on stems, other places, but that is basically like a plant tumor, and it apparently results from these infections with the bacteria. From my understanding, it doesn't usually kill the plant, but it is just kind of an aesthetic growth that's the consequence of infection. So this is a naturally occurring thing. The bacteria and the bacteria's ability to infect the plant is all straight out of Mother Nature. Nobody invented that. Simply, it was just discovered. So with that discovery, they found a mechanism to modify plants. So they realized, here's a naturally occurring entity that will transmit DNA into a plant. So once again, just like the bacterial plasmid, Mother Nature already has this transferable genetic element. So just like in the previous example, scientists figured out that they could modify that plasmid. They could cut out the DNA, or cut the plasmid DNA, the TI plasmid. And in the same way with bacteria, basically, they modify the plasmid to contain a new gene. And from there, it goes back to Mother Nature. You modify the plasmid, and then you allow the bacteria to interact with the plants as it does normally. Now, when I say normal, you allow this to take place in a lab under artificial conditions. So it's not quite normal, but in theory, it's a normal process by which the plasmid is transferred to the plant cell. The plant naturally uptakes the DNA. The difference, obviously, is now the DNA is different. So with this technique, you can add different genes into a plant genome. Now they do this, they actually start with plant cells in a petri dish. And then from there, they're actually able to get some of the first plants that grow out of that, believe it or not. And these create the modified organism. These grow into the modified organism. From there, you then do cloning, and they can actually create seeds from this mechanism. And then you can end up with genetically modified seeds that can be produced and commercially sold. So skipping past a lot of the detail there, but that is ultimately what's happening. Modify the plasmid. Plasmid is transferred to the plant cell. Plant cells now have modified DNA that is expressed, and that creates the GMO, which is then propagated through natural plant propagating mechanisms, such as what's called cloning, where they simply cut the plant and get it to grow new roots and create new plants from that mechanism. Okay, so we'll go through the steps again here. Gene of interest isolated, plasmid extracted, and cut. The gene of interest is added to a new plasmid, a TI plasmid, and ligases splice that together. That plasmid modified is now put back into the bacterium, the agrobacterium, and the agrobacterium is introduced to plant cells. Plant cells will be infected. Some will, some won't. The success rate is variable, but eventually you get some successful integration. And then those successful early cell cultures are grown into full-size adult plants, which are then propagated into many clones, which are then used to produce seeds. So ultimately, the discovery of the TI plasmid is what was the major breakthrough here, because Mother Nature already has the mechanism for this to take place. So the human alteration came from the way the plasmid was altered. Everything else, more or less, takes place due to or under natural circumstances. Now, the way that they modify the plants, or I should say the genes that are added, is what differs from one type of GMO to the next. And GMO plants, GMO crops for food production, tend to be the most controversial. No one seems to really care, from my understanding, about modified microbes, probably just because they're less aware of them. No one seems to really have an issue with that. Transgenic animals, certainly people probably have more of an ethical issue with, probably a nutritional one as well. But it seems like most of the debate revolves around GMO plants, from my experience. And there is some legitimacy to people's outrage. And so some of the first examples that they used this to make the types of GMOs they were making were plants that had built-in resistance to pesticides and herbicides. So they could actually add a gene, for example, that would make a plant resistant to an herbicide, which would be like a weed killer. And those were some of the first examples. And the downside of that is that they actually allowed farmers to increase the use of herbicides, like weed killer, Roundup as an example. So the GMO technology, the way it was used, was actually used in a way that actually encouraged pesticide and herbicide use. And that is what a lot of people are outraged about, and I think that's rightfully so. I think there is legitimacy. I don't think that increasing herbicide and pesticide use is a good thing, but really it has to do with the mechanism that they employed, the example that they employed, which was herbicide resistance. That in particular was what made that controversial. There are different ways that plants can be modified, and we'll talk about a few of those examples here in just a second. So what I was alluding to there a second ago, one of the first examples of a GMO produced on a mass scale certainly was called Roundup Ready soybeans. These were started in the early 90s, and by the end of the 1990s, around 2000, this particular GMO crop accounted for something like 90+% of soybeans that were being produced. So it was highly effective. So the way it worked is they took a gene that conferred resistance to a commonly used herbicide called glyphosate, more commonly known as Roundup. That's the commercial name, produced by a company called Monsanto, highly controversial company. So Monsanto makes Roundup, by the way. They make a lot of chemicals. They are a biotech company involved in genetic modification as well as all sorts of chemical engineering, herbicides, pesticides, etc. So they created both the Roundup Ready soybean and the herbicide that's used in conjunction with it. So normally you spray Roundup on a plant and it dies. It's a weed killer, but it has a pretty normally speaking, has a wide range of toxicity. Kills most plants. And so when it came to farming, if you ever tried to grow crops, you know that weeds are a constant issue. So de-weeding a garden, especially a large agricultural crop production, is extremely time consuming, and ultimately the weeds restrict the overall productivity of your farming. So with this invention, what they basically were able to do was confer resistance of the pesticide, the herbicide is what it's called, the Roundup, to the plant. And then you could spray an entire field with Roundup. And because the crops were resistant, it would only kill the weeds around the crops, and the plants would keep growing. So it basically allowed farmers to indiscriminately spray their field with Roundup and not have it affect the crops. And this was a pretty significant innovation for farming, because it allowed for just very easy manipulation of weeds. And so sometimes you'll see planes flying over a field, and one of the things they might be doing is spraying this chemical. Although there's lots of ways to apply it, so that's not necessarily a given. So the advantage to farmers was you had a very easy way to get rid of weeds, which were a constant problem for your crops. The downside is that it actually increased pretty significantly the amount of herbicide being used. So now it basically encouraged farmers to use more of this, because it had really a negligible effect on the actual crop itself. So people are rightfully concerned about this, because it increases the amount of pesticides in the environment. And just recently, in the last few years, there's been a growing concern about the safety of glyphosate. And if you just do a quick Google search on glyphosate cancer, pops up pretty quickly, and you'll find a lot of questions being raised about the safety of glyphosate. So I have yet to see anything confirmed, and sometimes it's very difficult to do, but there are definitely questions about this chemical. And so it being used widely, it being found in the water systems is a big concern, and that's rightfully so. So that's an example where people look at a genetically modified organism and they say, this is bad. And that's because the GMO increases pesticide and herbicide use, which is also bad. And I agree. In this example, I agree. I think it's a bad thing. I think it's good for the farmers. I think it's a situation where you don't have to have it. And while it is beneficial in some ways, I think the overall effect is negative. And I'm against this particular GMO. And I think that's the way people should look at GMOs. They should look at them on a case-by-case basis, and they should look at each one and determine whether it is good or bad based on the context. I look at GMOs a lot like I look at the example I use, I should say here. I look at them like a TV show. And a lot of people don't like one TV show or another for various reasons. It's violent, or it's got sex, or bad language, or you just think it has a bad message. A lot of people are against TV shows. But people treat GMOs like they would maybe a TV show, in that they want to get rid of all GMOs, kind of like someone saying, I don't like this TV show, therefore I want to ban TV. That's kind of how I look at the debate around GMOs. People say, there's some bad GMOs, let's get rid of GMOs. That's like saying there's a bad TV show, let's just ban TV. Well, that's not necessarily a rational argument. The more rational argument would be to ban that particular show, if you even agree with that. The difference with TV, obviously, is you don't have to watch TV. With food, we're more or less being forced to consume certain amounts of GMO food. So it's not a perfect analogy, but the idea is that you're kind of trying to throw the baby out with the bathwater, so to speak. So each example is different. Now, I've got another controversial example, and then what I consider to be a good example. So 2 bad ones and 1 good one here. Another bad example. This is one of the more dubious GMOs that people are against, and again, once again, I agree. So this is called BT corn. BT corn is an example where they've actually found a naturally occurring toxin to insects, and this comes from a bacteria called Bacillus. The B stands for Bacillus thuringiensis. So it's another soil bacteria that produces a natural insecticide, and I should say an insect toxin. So it turns out that these bacteria tend to propagate themselves around the roots of certain types of plants, and it's a symbiotic relationship with the plant that actually makes the plant resistant to certain insects because of the bacteria. So when the insect, usually worms and larvae and things like that in the soil, when they go to feed on the roots, the bacteria are there around the root protecting it, and when they eat the bacteria that are on the root, the bacteria have a toxin inside of them that kills the insect. So it's an interesting symbiotic relationship. So what scientists figured out was, here's the gene for that insect toxin called the BT toxin, and they were able to isolate that gene and insert it into a corn plant. So they literally inserted the gene into the entire corn genome. And now what you have is a corn plant that produces the insect toxin systemically. So this is different in that normally insecticides are sprayed onto the crop. So it's a topical application, and you spray it, and the plant is now coated in the insecticide, and any insect that feeds on it theoretically is killed. There's pros and cons to that too, by the way. Spraying insecticides is a lot of downfalls. It's less efficient. Lots of it gets into the environment. It gets caught in the wind. It goes all over the place. There's downsides to that too. So there's no real perfect way to do this, by the way. But in this scenario, what they're doing is they're basically growing the toxin inside the corn. So wherever the corn grows, the insecticide grows within it. So now if an insect goes to feed off that corn, it dies in a similar way by eating the corn plant. The problem with this is you can't wash the insecticide off. And now whenever you eat this corn, you're essentially eating that insect toxin within the corn. Now, all the research suggests, to be fair, the research suggests that the BT toxin is not harmful to people. There's been no evidence, from my understanding, that this insecticide, this insect toxin, has any sort of effect on people. So there's a thing called selective toxicity. Some things are only toxic to one organism and not to another. So in theory, this toxin is selectively toxic to insects and not to people. Now the problem is that research when it comes to toxins is never really good enough, in my opinion. It's always questionable as to whether the research was sufficient. You're not talking about research on people. They don't do long-term studies. When they say it's safe, that means there's no obvious short-term effects in test animals. And the big question is, well, what about subtle long-term effects in humans, which is what matters? And really the research isn't good enough or sufficient enough to ever really state that that is the case. So you can always question, and I think people should always question, the safety of these studies and the legitimacy of the long-term aspects. And so once again, I'm against this particular example. I don't think the research is safe enough or good enough to legitimize the use of these types of the use of an insecticide in this kind of way. Now here's what's ironic about it though. You can find quote unquote naturally occurring insecticides that have the same thing, that if you spray them on the plant, people consider that to be somewhat of an organic mechanism of using an insecticide. So here's an example of an insecticide with the Bt toxin that would be sprayed on a plant as opposed to grown within it. So the truth is, farming is difficult. If you've ever tried to grow a garden, one of the things you've probably found is that between the weeds and the insects, if you don't do something about them, they'll pretty much devastate your crops. It depends on the location, the year, the crop itself. But if you don't control insects and you don't control weeds, you're going to lose a lot of crop, and you're going to have the very least a smaller yield than you would otherwise. And in some cases, they may take all of it. And then fungi are another issue on top of that. So between fungi, weeds, and insects, if you don't do something about it, you're just going to have all sorts of issues. So the truth is, when it comes to farming, you have to do something. And when it comes to making crops cheap and affordable on a large scale production level, you have to do something that's cost effective. It would be ideal if every farm could pay people to go pull the weeds by hand and control the insects as organically as possible, using the fewest amount of pesticides and just kind of evasive type methods to prevent them from coming in. But that's just not realistic. And when you do that, like when you have organic crops, that dramatically raises the cost. If you look at the cost of organic food versus non-organic food, it's oftentimes twice the price, maybe more. And a lot of that has to do with the fact that it is more expensive to grow these crops without these interventional chemical uses like herbicides and insecticides used in a more effective way. So that is ultimately why organic food is so much more expensive. And it's definitely controversial to use these things, but when you look at why they use them, that is why. It's also important to note that it's not totally different from some of the quote-unquote organic methods that might use a similar chemical but spray it on the crop as opposed to grow it with it. And I think that is better because, again, you can wash that off, but it's not entirely different. So it's a different delivery mechanism. Now, this last example here is not like the only example of these are not the only three examples of GMO plants, but this is an interesting example that I think gives you an idea of a positive use of GMO technology. So this is an example called golden rice. And golden rice is a rice plant that has been engineered to contain a precursor to vitamin A. It says contain vitamin A. It actually contains the precursor to vitamin A, which is a chemical called beta-carotene. So rice normally does not contain this vitamin or the precursor to it. And for a lot of people around the world, rice is a staple part of their diet, and they end up eating a large amount of calories from rice. In especially parts of Southeastern Asia, there's not a lot of vitamins in poorer parts of those regions. And because the main food that they're eating doesn't have vitamin A and they don't get lots of other sources, a lot of people end up being vitamin A deficient, especially children. And this is a big problem because vitamin A deficiencies, especially at a young age, can lead to blindness, and it can lead to all sorts of vision disorders. So in these areas of the world, it's not uncommon for people to develop blindness or severe vision disorders, and it results more or less from a lack of vitamin A. So a simple solution is to provide vitamin A in some form or another, and as a result, you can prevent blindness and other vision disorders. So this is an example where, in theory, you're actually making a plant more nutritious and adding a naturally occurring vitamin to a plant that just doesn't normally have it. Now, you could argue that that's playing God and that people shouldn't be manipulating Mother Nature. And I think if you take a hardline stance that there should be no manipulation, then it's hard to argue that standpoint. But if you're looking at pros and cons, to me, this is a pro. And the pro here is that you're making the food better. Again, that's an opinion, but it is definitely a deviation from these other examples where you're making the food more effective in reference to the use of an insecticide or herbicide. So, in my opinion, these are examples of what I would consider controversial at best and arguably bad GMOs. And this is an example of one that has a great deal of beneficial potential. So this is a scenario where when you're looking, oops, I'm sorry, I passed that slide there. Sorry. I was trying to finish up on golden rice here. This is an example where the golden rice is a benefit and less of a detriment. Now I will show a video at the very end where some people actually don't agree with me on that, and there's actually a controversy around this, not so much for the benefit of the vitamin, but more from the controversy of the corporate use of it and how that might affect the farmers and their finances in reference to it. So this is actually controversial. Okay, so back to where I left off here. These are some of the pros and cons of transgenic plants. Some of the pros are that you can ultimately create higher yields, and the higher yields come from better success rates. Fewer weeds means bigger crops, theoretically. Fewer insects means more crops that are actually commercially viable. Most people don't want to buy apples, for example, with a great big hole in them. So theoretically, you can produce a higher yield because you tend to get a better return. A higher percentage of the crops that are grown actually make it to the commercial market. Crops are being altered in ways that allow them to grow in new locations. Drought resistant, salt resistant, even flood resistant crops that can withstand standing water. So these are strains that are currently in development that one day could be used to grow in new locations, and with the impending impact of climate change, it's widely accepted that the location of crops will have to change. We won't be able to grow crops in the same places that we always have traditionally. And some of that may be growing them in places where they weren't previously grown at all. And that's a potential that could come out of this. Now, to be fair, you could also do that by using non-GMO mechanisms. Selective breeding can actually create the same effect, theoretically. So to be fair, you wouldn't necessarily have to use GMOs to do this, but it's a more effective way of doing to getting to that end point, which is to make crops grow in new locations. And as I mentioned with the last example there, golden rice, crops can be modified to contain vitamins. This can be extended to all sorts of endpoints here. They've actually looked at making crops that can contain vaccines within them. Here's an article from Scientific American talking about edible vaccines and the potential for one day to actually confer an immunization through the food. People are going to obviously flip out about that. Oh my God, the government's contaminating our food. And again, this is questionable, no doubt about it. But you could argue that this is a good thing in that you're going to get a vaccination through a more natural way, maybe not natural, that's probably not fair to say, but from a more effective way by growing it into the food. So a lot of people are just going to be against any sort of idea like that whatsoever. But again, debatably, these are potentially positive mechanisms that a GMO could bring. So once again, if you're against the whole manipulation from any viewpoint, then it's hard to argue any pro side of GMO. But if you're simply looking at GMOs as a bad thing, what you really need to understand, as I mentioned before, is that each example is different. Okay, so as a general negative side of GMOs, where a lot of people will say that they are problematic, comes from the fact that we really don't know the long-term effects of the GMOs. The safety studies are done on test animals over relatively short periods of time, if they're done at all, and really the research is never really conclusive to say that something is definitively safe. You take the BT corn, as I already mentioned. Allegedly, that's safe to consume, but no one's ever tested the human species for 50 years. No one's ever had a group of people, for example, that were fed BT corn every day for 50 years and then studied them to see what kind of effects they might have compared to a group of people that weren't fed that corn. You don't ever do those kind of studies, though. And the truth is, that's the case when it comes to every single questionable chemical product or, piece of technology. People wonder about cell phones and the waves that they create. Wi-Fi signals maybe interfering with your brain. None of these things are ever deemed to be 100% safe. Not a single one. The research is always questionable. It's never good enough. And at a certain point, you do what research is accepted, what is mandatory, and then you roll the product out. And that's the way it works. And I have all sorts of issues with that. But the fact is, it's not just GMOs, it's everything. So it also applies to GMOs, but it's not just GMOs. So when it comes to the long-term effects, you could say, yes, that's the case with GMOs. I would say that's the case with everything. And so I don't think it's really that much different than the safety efficacy about the paint on your walls, or the hairspray that you use, or the makeup that you use, or the fingernail polish, or whatever it is. Also the bug spray that you use. All these things have limited safety, limited studies, that test their safety. And there's always questions about how safe they really are. And in GMOs, the context is a little different, but it's basically the same scenario. So I don't really consider that to be the best argument against it. I think you could argue that against every single product that's being sold. So they do test to an extent, and it's like most products, that is, they go through the different testing agencies. The EPA looks at the safety on the environment. The FDA looks at the safety for the consumer, and the USDA often looks for the safety of other animals and the ecosystems around it, and how that relates to some of the economy and farmers and things like that. So each one of these agencies has a different perspective on the quality and safety of the product. The number one thing about these, though, is that you have to really question the efficiency. And kind of as I was alluding to a second ago there, none of these studies, in my opinion, are ever really good enough to say that something is definitely safe or definitely okay for long-term use. So I question every single one of these agencies and ultimately the data that they provide. I think they do the best they can. I don't think that they intentionally lie. I don't think that they intentionally manipulate. I think there maybe are exceptions to that. Maybe there's some cases where they do. But as a general rule, I think that they do the best they can. But the ability to determine something is 100% safe, I would say, is nearly impossible. So those are some of the issues. In my opinion, the real issue around GMOs lies in the fact that almost all of them, I think it's 100% of GMOs, come from about 5 companies that are all major corporations. And when you understand the way a corporation functions, you understand the major issues that any corporate decision making process involves. So the corporations themselves have a sort of logic that, in my opinion, is very harmful because they're always based on profit, and they've been proven to mitigate human safety in the interests of profits. This is many examples. I won't even get into that. Be here all day. So, in my opinion, when you're worried about GMOs, you should be more worried about the fact that it is corporations that control them. The technology is what you want it to be. If you want to make a vitamin, you can make a vitamin in your plant. If you want to make the plant have an insecticide, you can do that. And you can argue that one is good and one is bad. But they're both a GMO, and they both are being downplayed by anti-GMO activists who want to get rid of both of them. And you can certainly argue that one is good and one is bad. So, in my opinion, it should be looked at on a case-by-case basis. Now, one of the major undeniable issues with transgenic plants in particular, is that they will interbreed with wild-type species. Some strains more so than others. But undeniably, when you add a gene to a plant and then you release that plant into the wild, some of the genes are going to get mixed in with the wild type. And this is not even debatable, it's been definitively shown to happen. So this is a very real issue with this sort of technology. So what ends up happening, like if you take the example of Roundup Ready soybeans, where they use a pesticide or herbicide, within the plant, what ends up happening is the herbicide gene gets transferred to the weeds, and the weeds end up gaining the very gene that makes them resistant. And so ironically, the gene that is designed to prevent or that is designed to increase herbicide resistance ends up getting into the very weeds that it's designed to kill, or designed to target, I should say. So ultimately, the more you use this type of transgenic plant, the more it spreads to the wild type plants, the weeds, so to speak. And you end up actually in a scenario where the product stops working as effectively. So then you have to create a new GMO, and you have to create a new pesticide or herbicide because the old one wasn't working. And it's kind of like antibiotics. Just like the bacteria develop resistance over time, so too do the weeds. And the issue then is that you now have to have a new chemical that the weeds aren't resistant to. So then you start to use that. Well, then they develop resistance to that one too. And just like the antibiotics, when you start using it, the organisms in the environment, they adapt and they become resistant over time. And it seems like an unavoidable consequence. It doesn't seem like something that humans have a way of getting around. So that's probably one of the more legitimate arguments, is that you are actually creating hybrid species in the environment that end up taking on the genes that are put into these GMOs, and they end up spreading throughout the environment. And that's a real issue, and I think it's an unfortunate, inevitable side effect of this technology. So whether that's enough to condemn it or not is an opinion. I personally don't think that it is, but I definitely think it's an issue. And I think if people want to be realistic about the objective problems, that is definitely one of them. And by the way, the idea that GMOs cause cancer and they cause all sorts of health effects, that's not proven. There's no real science that shows that is a fact. It is nothing but conjecture. Now, it's good to question it. As I mentioned, you have to question the safety studies themselves. But as far as proof that they cause harm, there is none. So that's all speculation. But there is proof that they do cause the transfer of genes to wild-type plants. That is not speculation. That is definitely true. Okay, and the last part, really, when it comes to, like I mentioned, when it comes to these GMOs, in my opinion, the biggest issue is it's the corporations that are the problem, and it's the way that they use the technology, not so much the technology itself. The technology is like a TV show. You can make a good TV show. You can make a bad TV show. You can make a family-friendly TV show. You can make a TV show full of smut, violence, and filth. It just depends on the producer and what they want to do with their platform. The GMO is very similar. You can modify a plant in a way that's good, or you can modify it in a way that's controversial and bad. It just depends on what your goal in this case, from a corporation is. And because corporations are dead set on profits, they're more concerned about increasing the yield of the crops. They're more concerned about making the crops more resistant to insects and weed killers and things like that. And that comes with some potential side effects, some consequences because of that intention that we as consumers are concerned about, rightfully so. Okay, so here's an example, going back to the golden rice, where I said the golden rice is a good example in my opinion, but there's a group of activists here that are against it because they see it as a kind of a corporate takeover of the rice production in this part of the country, this part of the world where the rice is being grown. So I'll watch this video and that'll be the last part of this section.
Angry farmers tear down a fence and destroy an experimental rice paddy in the Philippines. They say the genetically modified rice could contaminate their own crops and put them out of business. But scientists at the International Rice Research Institute believe their golden rice will provide millions in the developing world with a much richer diet. Institute spokesman Bruce Tolentino says the rice is genetically modified with beta-carotene, which boosts its vitamin A content.
In vitamin A enriched rice, what the scientists did was to select 3 genes out of roughly 30,000 genes in a rice plant. They just took 3 genes and transferred those from corn into rice. You cannot do that with traditional breeding, but with advanced biotechnology, you can do that.
The Institute says golden rice is still under development, but that local authorities are gathering data for submission to government regulators who will assess its safety and value as a food crop. Trixie Concepcion from Earth Island Institute says there's plenty of options to fight malnourishment. Golden rice, she says, is about nothing more than corporate profit.
There is no need for this expensive science and this expensive product brought about by corporate science, which we believe will not benefit the local farmers. It will not benefit those who are interested in healthy food. It will not benefit the community as a whole. It will only benefit the corporations who are pushing for this product. And that is the reason why they're pushing this very hard on third world countries.
Critics also say that golden rice and other bioengineered foods are dangerous because the long-term effects of genetic modification have not been fully investigated. But Tolentino calls those criticisms baseless. He says there's an immediate need to lower the risk of blindness and malnutrition for the estimated 1.7 million Filipino children currently suffering from vitamin A deficiency. And golden rice, he says, is a safe and effective way to do it.
Alright, I apologize that was so choppy. That was the best I could do. I apologize. So anyway, that ends section 10.2 here. So as always, email me with any questions or bring those questions to class. Have a great day, and I'll see you next time we meet.